Good day, everyone. My name is Arvind Varsani, and I'm going to be talking to you a little bit about virus taxonomy and virus taxonomy, particularly in a data-rich world. So virus genomes are kind of weird in the sense that viruses can have a variety of different genome types, ranging from double-stranded DNA genomes all the way to RNA genomes. Now, these double-stranded DNA genomes can be linear or circular. They can also have single-stranded DNA genomes, which are either circular or linear. Viruses that have RNA genomes come in different forms. They come in those that have double-stranded RNA genomes and others that have single-stranded RNA genomes that have different polarity, either the positive or the negative sets. Then we have groups of viruses that integrate into the chromosomes. Here are RNA viruses, mainly retrotranscribing viruses. So these are retroviruses such as HIV. These are RNA viruses, single-stranded RNA viruses that integrate into the chromosome of their host. And we also have double-stranded DNA viruses that integrate into the chromosome of their host. So this could be very analogous to what the bacteriologist would kind of look at in terms of temperate phages that actually can go through a lytic or lysogenic cycle, and the integration is the lysogenic cycle to form a prophage. Here they form a provirus. There are no universal genes in viruses. So unlike eukaryotes and prokaryotes and archaea, what you have is you have certain genes that are conserved evolutionarily, mainly the ribosomal RNA genes. So with that, you could actually use these kind of information or sequence information derived from these ribosomal RNA genes to form operational taxonomic unit and do comparative analysis. When it comes to viruses, we do not have any of these. There are certain groups of viruses that have certain genes that are conserved, but that's about it. Not across the entire group of viruses can you find a single gene that is conserved. So with that, the taxonomy of viruses becomes a lot more complicated because you can't use one model fits all. And with that, there is a lot of variation in taxonomy of viruses. Now, beyond this, the other thing that we need to think about is evolution rates, because these also affect taxonomy in a greater scale of things. So viruses use three different strategies to evolve. Basic substitutions. Now here, if you look at the substitution rate of viruses, it's staggering in the sense that it's really, really fast. And RNA viruses generally use RNA-dependent RNA polymerases that are error-prone, and as a result, they have high substitution rates. But we're finding that there are DNA viruses that use host polymerases that have very similar substitution rates as well. And then there are other viruses that have variable substitution rates. Beyond this, what also happens during a co-infection is that if there are two homologous genomes in there, during replication, template swapping can take place. And as a result, there is generation of chimeric molecules or chimeric genomes. And this is one really useful way to repair mutations, but it's also a very, very unique way to explore sequence space very rapidly. And you can imagine that if you're using only pairwise identity-based indexes to try and discriminate or delineate viruses at a taxonomic level, you can get very, very rapidly new species emerging just through mechanisms of recombination. And also what you have is there are certain viruses that are either segmented or multipartite. And when they co-infect the host, they can reassort their segments. And as a result of it, you get these variable reassortants that come out and can complicate taxonomy. Now, when we look at viruses across the scale, there is no single thing that fits all in terms of their sizes, in terms of their morphology, their variable sizes and shapes. And when it comes to the genomes, they go anything from 1600 nucleotides all the way to megabases of genome size. So their gene content is variable as well. Now, what has happened over the last decade and a half is primarily the sequencing technologies have changed, and they have actually enabled us to get really, really large amounts of data from uncultivated organisms. And as a result of that, we're discovering large amounts of new viruses. And it's primarily as a result of the second and third generation of sequencing platforms that have resulted in this. So with that, the virus taxonomy has been inundated with large amounts of sequence data that have been derived from metagenomic studies. And with that, we've had to come up with a plan to actually incorporate sequences that are derived from metagenomic studies into a virology framework, taxonomy framework. So if you go back historically and look at how viruses were classified, they were mainly classified based on their biology, which was either in vitro properties, virion structure, or antigenic properties, and obviously host range factors from pathogenicity all the way into vectors and other things. And then we went to an era where sequence information came in, and with that, phylogeny was incorporated. And we're in this era here where we're basically now gone away from biological data only to pretty much incorporating sequence data, where we think sequence data has a lot more information than just biological traits. Furthermore, what also has happened is that we've gone from a 5-rank structure in terms of classification to a 15-rank structure, which helps start establishing much more deeper evolutionary relationships as well as taxonomy classification. So here's an example of how this would look in an era where you have Ebola virus as an example. It's a species, Zaire Ebola virus, and how that's structured. It's in the realm Riboviria, in a kingdom, in a particular phylum, with a subphylum, particular class and order, and then it fits into a family called the Filoviridae, and then in the genus Ebola virus. Now, as I mentioned earlier, that the sizes of the genomes vary. There is not a single gene that is universal across all viruses. It is not surprising that there is no single solution for virus classification. And also, given that they're evolving at different rates, the taxonomy of viruses has to be dynamic. It cannot be static. So with that, if you look at it from that perspective, that each group of viruses will then have its own species demarcation criteria. And within that, there are problems as well. But I'll give you some examples in terms of what are the different variations and how there are advantages as well. For example, papillomaviruses have roughly 7 kilobase genomes. They're classified based on one single protein with a species demarcation threshold of 70%. So anything that is less than 70%, similar to anything that is known, would highly likely be a new species as long as there is good phylogenetic support for it. When it comes to circoviruses that have roughly 2 kilobase genomes, their criteria is based on full genome pairwise identities with a threshold of 80% coupled with phylogenetic support. Gemini viruses, these are roughly 3 kilobase genomes, but here the classification varies based on different genera within the family Geminiviridae, ranging from 78% to 91%, all based on full genome pairwise identity and phylogenetic support, but it's very genera specific. When we look at double-stranded DNA bacteriophages with a huge range in terms of size, genome size, they're mainly classified based on pairwise identity thresholds with 95% threshold coupled with phylogenetic support. So you can see the variation across the board. And so that means there is no universal way of classifying all the viruses, no universal species demarcation criteria that can also be used in this setting. And also when it comes to naming the species, there's variations. And here are some examples of different variations that are there in virus species names, ranging from something like potato virus X, which is a host name with the virus, and then a letter. Then we have place names with a genus. So there is no universal format here. And with that, a proposal has been put forward to the community, which is one of these 3 options. We go into a binomial format where we can either have the epithet to be Latinized or Latin, alphanumerical, or a free form. And with consultation with the community, it has come out that the community is not in favor of genus coupled with Latin or Latinized epithet. So basically, it is highly likely that the 2 and number 3 options are going to move forward. And so future species names are going to incorporate these kind of setup, as well as the ones that are currently classified will have to be revised to conform to this nomenclature beyond this. And so basically the ICTV, which is the International Committee for the Taxonomy of Viruses, is a group of volunteers who come together and are associated with trying to come up with these kind of classifications. And they have subcommittees that deal with different groups of viruses. And within that, there are study groups that look at particular viral families. And so that means that there are experts dealing with each and every particular group of viruses, then proposing the taxonomy for it, and that gets reviewed and then ratified. So if any one of you wants more information on how the taxonomy of viruses is structured, please go to the ICTV website. There's a lot of information there. With that, I thank you all for listening, and I hope to have a very fruitful discussion with you in the panel session. Thank you very much.